Okay, so it's great to be talking to actual people and not a Zoom screen after 2 years of being at home and giving talks through Zoom. So I'm Marianna. I will talk to you about our work at IBM Research in Zurich on how we try to understand cancer heterogeneity. And to start, okay, so biological systems are inherently stochastic systems. They consist of a number of different entities. So we have DNA, mRNA, proteins, and so on. And now these components, they interact in a complicated way that's not deterministic. It also contains a very important stochastic component, as you can see here. So this stochasticity, however, is a fundamental property of these systems. It's not a bad thing. It's what allows them to respond to stimuli, it's what allows them to differentiate, what allows them to adapt to the environment and ultimately survive and thrive in their environments. Now, however, in cancer, the problem is that the normal cell proliferation is disturbed. So all the normal mechanisms that safeguard how a cell proliferates are disturbed. So what happens is that this heterogeneity, this stochasticity, is further amplified, and it leads to what we commonly refer to as tumor heterogeneity. Now, this is again something that we observe across all levels of a tumor's organization, starting from what happens in the inner workings of a single tumor cell, to a whole tumor, to a patient, and to a whole population of patients. And of course, it contains intrinsic sources, starting from, of course, genomic instability. Now, this instability is propagated in all the different biological entities, so the epigenome, transcriptome, and so on, but it's also further amplified by other sources of stochasticity at all these levels. And of course, a very important component of this stochasticity concerns the tumor microenvironment. So it's not just what the single cell is doing, like intrinsically, but it's also where it is, right? And it has a very important temporal component, so how do these cells proliferate when the tumor grows, but it also has a very important spatial component, and this is something you can observe in the topology of the tumor ecosystem. So now, in the last years, we've been really working on that and trying to understand how this stochasticity occurs and how it's regulated and how it's linked with disease initiation and progression and so on. And we've done this across all levels of a tumor organization, let's say, starting again from the inner workings of a single cell. And something that here is very important that we found out the last, I would say, 10 years, is how exactly the genome is folded within the nucleus. So we now know that this is something that is linked to the regulation of gene expression, and it's also tightly linked. So disruptions of this thing are tightly linked also to cancer, right? So in the past years, we've worked a little bit on that, and here you see one example of a machine learning model that we developed that, without going into a lot of details, I'm happy to talk about it later with you, but essentially it takes as input genomic data, and it uses this model architecture that you see here, so a seq-to-seq model, to encode how the genome is actually folded in 3 dimensions. And this we do not only to understand the temporal pattern, the pattern, but also to see how this changes over time, right? So if you have time points, you can actually simulate how the folding changes at different time points. But what is even more exciting is starting looking into populations of cells that form a tumor, as what you can see here. And another project that we worked on here, we focused more on temporal heterogeneity, so understanding the temporal dynamics of heterogeneity is called cell cycle tracer. This was, again, a study we did a couple of years ago, where what we had, again, very briefly, was a collection of single cells. And what we try to understand is how does their cell cycle, so how does their lifespan affect how they react, and very importantly, how also do they react in drug perturbations. So what we found through that is that actually cells tend to react differently at different stages of the cell cycle. So if you would perturb them with an inhibitor or some chemical substance, you would see that how they would react would not be the same, right? But what is very interesting is now how all these things are linked to disease at the inter and intra-patient level. And to further understand that, in the past years, again, we worked in a tight collaboration with the Bodenmiller Lab from the University of Zurich in what we call a single-cell atlas of the breast cancer ecosystem. So now, in this project, we had a large collection of different samples from different patients, and for each patient, we used suspension-based mass cytometry to quantify a total of up to 26 million single cells. And these were cells of the tumor compartment, but also of the immune system. So what we were very interested to understand here is what are all these different cell populations, tumor and immune, how do they interact, how abnormal they are, and how can we use this type of ecosystem, as we call it, analysis, to understand and better stratify the patients. And again, I don't have a lot of time to tell you like all the details, but the main idea of this approach is that we saw that this ecosystem-based approach, so looking into networks of cells and not only looking at unique biomarkers, was able to show us that indeed, for example, a certain category of patients might be good candidates for immunotherapy and so on, so to enable patient stratification. But we also were able to devise some, as we call, heterogeneity scores. Now, these are scores that are learned from the data, so data-driven scores, and through those, we were able to show that the tumors that were very aggressive were actually consisted of a few cell phenotypes, but those phenotypes were very, very abnormal, and they were very unique. So occasionally, we might even have them only in this specific patient in the big cohort that we had. Okay, so now all this is very interesting, but it obscures one very important level of regulation, and this is how the tumor is organized spatially. So tumors are spatially heterogeneous ecosystems, right? So it's very important to actually be able to observe which population is next to which. And this is one example of a ductal carcinoma that you see here, right? And you see you have a lot of normal cells, but you also slowly have the presence of multiple different abnormal, so tumor cells, and also immune cells from the microenvironment. And then maybe many of you are familiar with these famous hallmarks of cancer. So this is a review paper, I think one of the most highly cited review papers in the history, some tens of thousands of citations that has been revised now in 2022. And what it shows here is essentially what are the main components, the main mechanisms that contribute to tumor heterogeneity, and specifically to spatial tumor heterogeneity. So what we are able to understand here is that the interactions of all these different cell types, so exactly where they reside on the tumor ecosystem, is very important, as it can lead to the emergence of a number of different hallmarks from the ones that you see. So for example, if you're next to a cell that secretes a growth factor, you might also absorb this growth factor and become more proliferative and so on. So it's very important who is your neighbor, right? So, however, other interactions are also of great interest. And of course, most important would be the interaction between tumor cells and immune cells. And of course, what you would normally get is that immune cells would attack the tumor cells and would kill them. But unfortunately, this is not sometimes the case. Sometimes the tumor cells are managed to trick the immune system and evade apoptosis, right, from the immune system. So when this happens, it's also very important for immunotherapy selection and so on. And lastly, even the topology of the tumor itself is very important, because how the blood vessels are organized and the vasculature is organized would lead to variable access to nutrients, growth factors from the bloodstream, or to regional hypoxia and so on. And this is again a very, very important contributing factor to this very chaotic environment of the tumor ecosystem. So of course, this is AI and pharma, so we're very interested to understand, how can we actually explore this, and what is the role of different treatment pressure. So different treatment pressure also is very important, because it could lead to the emergence of resistant clones. So some cells, through this whole stochasticity, might learn to acquire resistance to a certain drug and still manage to proliferate, grow, metastasize, and so on, which is what we want to avoid. And of course, what is very important is that all these different mechanisms that I'm showing you in the past years are becoming targets for treatment. So the tumor microenvironment as a whole is now a druggable entity. So some examples of drugs, of course, maybe the most, I would say, famous example is immunotherapy, right? It targets these types of immune and tumor interactions, but also other drugs try to kill off the vasculature, for example, and there are multiple different mechanisms where we can give different drugs to modulate not target and kill the tumor cells themselves, but the microenvironment that enables their growth, let's say. Okay, so of course, as you understand, we're very, very interested in deconvolving this huge complexity within the tumor microenvironment, understand what are these populations, how do they interact with each other, where are they found in space, how do they manage to evade immune destruction, and how does the TME ultimately cultivate the tumor? How does it enable the tumor to grow and possibly even metastasize? And if you view it like this, then you can actually view the tumor microenvironment and the stochasticity within the tumor microenvironment as an opportunity for new treatment, right? As an opportunity for new biomarker discovery. So then heterogeneity actually becomes an opportunity. So along these lines, what you need, of course, is data, right? There's no machine learning without data, and you need good data. And in the last years, what has been a great revolution in this field is the emergence of spatial omics, usually single cell, but not necessarily single cell. And they are broadly categorized in two big groups, spatial transcriptomics and spatial proteomics. And of course, they differ in what they quantify. So transcriptomics, of course, mRNA, and proteomics, proteins, but they're also very different in their properties. So for example, within the spatial transcriptomics field, you can have fish-based or sequencing-based approach. These would be quantifying a lot of single cells within a probe, like 10 single cells. So they're not exactly single cell approaches, but still they offer very good resolution, whereas these ones are actually single cell approaches. And then with spatial proteomics, what is very exciting is that on the one hand, you cannot quantify a lot of proteins. So it's a panel-based approach. So you would get from a dozen to 40, 50 proteins. But at the same time, because you're looking at proteins, you know exactly what they're doing. So there is a gap between mRNA levels and protein levels, but with the proteomic measurements, you have much better insight about the function, right? So in the past year, we've been working on developing a framework to actually do exactly that we call Athena. I hope you like the logo. And essentially, Athena is a way that we ingest this data, these spatial omics data sets. So what we need for Athena to work would be the raw multiplexed imaging data and the segmentation mask, so we don't do the segmentation ourselves. And then given a phenotype for each single cell, what Athena does is first create a graph representation of the tissue, and then quantify spatial heterogeneity with a number of different scores. And ultimately, if you have this representation of the tumor, you can analyze your data. So if you have a big cohort, for example, you can do survival analysis, statistical analysis, and so on. But you can just simply explore your data, let's say, if you don't have clinical metadata. So Athena is the result of the very hard work of Adriano that you see here. And we have it open sourced on our team's GitHub, so please go ahead and check it out if you're interested. And just very briefly take you through exactly the framework and what we're doing. So we start with multiplexed imaging. Now for this example, I will be showing you image and mass cytometry data from a recent publication where the authors, what they did is quantify a breast cancer cohort that you see here with an antibody mix. So it's a TMA that was stained with a different antibody mix to quantify approximately 36 proteins, I think. And this would look like this. So for exactly on the same tissue section, you would have, let's say, quantification of different proteins, right? So how much does each single cell express this protein? And then if you have this information, you can actually explore it to assign a cell type to the cell. So whether it's a tumor cell, a myoepithelial cell, an immune cell, and so on. But exactly because you have a lot of data, you have like 40 different features, and these are selected, right? You could even do a much more detailed phenotyping, as we say. So what type of immune cell? Is it a T cell or a B cell? Or what type of tumor cell, and so on. So you get to these very interesting ecosystems, right? So as I told you before, what Athena does is first represent the tumor as a graph. And here we use different graph building approaches. One would be a radius graph, where you simply connect the cells that are in one specified predetermined radius. And this simulates how cells communicate via paracrine signaling. Another option would be a contact graph, where it's self-explanatory. You would join cells if they're touching, and this would simulate a juxtacrine signaling. But of course, just for to play around, we also have a k-nearest neighbor graph, because at the end of the day, you always need a k-nearest neighbor graph. So essentially, what you have now is this representation of the tumor here. You have your graph, and you have for each node an annotation, a discrete annotation, but you also have the quantitative data. So you know exactly how much, so a vector of expression for each different cell. And what you can do with that, what we do with Athena, is exploit different scores that I mentioned before, such as, for example, we compute the local entropy of all these neighborhoods to see where do we have regions that are very rich in interactions between cells of very diverse phenotypes. And as you can see here, for example, like the tumor immune border is very nicely highlighted. So all these tumors here, all these cells here in the middle are different types of tumor cells. You can see them here. They express different hormone receptors, cytokeratins, and so on. And all the green ones here are immune cells. And you would see them like nicely highlighted. You could also quantify the infiltration. So where do I have unique immune cells embedded, penetrated deep within the tumor mass, such as what you see here. you can quantify the interactions between different cell types, right? So which attract or avoid each other within this ecosystem. And you see again here, for example, like in this column, that all these reddish cells are avoided by most green and blue cells, but are attracted by other cancer cell types. So in this way, you quantify how strong, what is the pattern of interaction within the tumor. But you can also quantify things like how are those cells organized in space. So this is a type of analysis where a peak would indicate clustering, such as, for example, for these like purple cells here, right? And then if the curve is on the zero axis, it would indicate like random pattern. So with all this way, actually what you can get is you start like with your tumor that you have here, okay? And then you ingest this information and you represent it from a very, very complex image into a collection of interpretable features that tell you that's how much this population interacts with this population, or these cells are scattered, whereas in this case they're clustered, and so on, right? So you fall in a lower dimensional space, and then, however, the question here becomes, how can we actually use this, right? How can we use this to understand, what drug might be the optimal treatment for each of these patients? So very briefly, I will talk about two projects that we just kicked off a couple of months ago. The first one is called Promotex. It's a collaboration between our team at IBM and the spatial metabolomics team at EMBL in Heidelberg from Theodore Alexandrov, and the Eurogenous lab in the University of Bern, led by another Marianna. And then essentially the idea behind Promotex, it's about prostate cancer heterogeneity. And the idea is to use prostate cancer organoids. Now, these organoids are treated with different drugs and then profiled by spatial single-cell metabolomics that you see here. So these are our data. And now all these rich data before and after drug perturbations, we use them to train different models and so on. But ultimately, our vision is that we are able to create what we call a perturbation atlas of metabolic states. So if you assume that you have a non-cancer case and a cancer case, if I give like a different drug, where will my cells move into this space? You can imagine this as an embedding, right, of the cells. And then actually the question becomes, to identify the best treatment, what you need to do is, given the cancer data, you would need to find the drug that actually is able to shift the cells back closer to the non-cancer case, right? So this is like the main idea. But I don't know if it will work. We just started working on that, but I think it's a promising direction, let's say. And the second project is called Aries, and again, it's about prostate cancer. We have a prostate cancer cohort that you see here through our collaboration with Bern, with a clinic there. And then what we're doing is that we're profiling a TMA of these patients with imaging mass cytometry. Now, this is a little bit different. It's not metabolomics anymore, it's proteomics, but still it's along the same lines. You have an image of the tumor, you perturb it ex vivo with a drug, and then you see how the spatial composition of the tumor changes. So maybe some cells will die, some cells will move, and so on. So you basically have a graph in space, right? And ultimately, the goal, the vision in this project is that by exploiting these types of techniques from graph representation learning, what we can do is, given an image of a patient before the drug, we can actually simulate how the tumor would look like after the treatment. So perform in silico perturbation, drug perturbation. And of course, if we're able to do that, it's like really nice, because you can apply it also to paraffin-embedded tissue, not just in live tissue, and then you can actually have a more personalized system where you can say, okay, this patient that has these genetics and this image would need to take this drug so that they respond better to the treatment. And with that, I think I'm just on time. Just to thank a couple of people. So most of the work that I showed you on Athena was by Adriano Martinelli, and a lot of the work on graph representation learning is led by our very brilliant postdoc, Pushpak Pati. A lot of people within IBM, but also from our collaborators that have been instrumental in this work in the past years. And as I mentioned before, please go ahead and check our GitHub page. But also, if you're interested in more general graph representation learning and digital pathology, please check out also histocartography. That is a library for that we're also very happy to be a part of. And with that, thank you. I don't know if we have time for questions.
I think we'll set up the... Give it a try to the Zoom with Patrick. So in the meanwhile, I think you could take a few questions.
Okay, great. Yes, I can stand here when you guys. Okay.
So yeah.
or you can just find me. Oh, okay, great.
I would have a question. So, how well can you actually determine within a tumor these spatial aspects? So, I imagine you take a biopsy. Normally, you get a very small subsample. How do you envision to get this clear picture of the biological standpoint?
Yeah, that's a very good question. So one of the biggest limitations is the availability of the tissue, right? So one approach would be you take different cores from multiple locations and you do a TMA approach where you have taken just all these different cores and you hope that this is enough to capture the heterogeneity, because it's very important where you sample in the tumor. So in the projects that I showed, we're using 4 different regions of the prostate, for example, but ideally what you would want to do is even more sample availability. But this is of course very hard because, for example, like these patients undergo radical prostatectomy, so the Institute of Pathology also needs a lot of material for diagnostics, for their standard diagnostics, right? So, and this is for research purposes, so of course we cannot, they want to do all the traditional imaging that they do and so on, and we want to be able to compare with that as well. So if the pathologist says, okay, this is tumor subtype, whatever, or Gleason score or whatever, we need to be able to, let's say, have an agreement or, compare. So you cannot ask for too much, but ultimately I would say that what we would hope would be like a whole slide image or something like this, that we can have a more detailed information of the tumor. But certainly it's one limitation.